lands of the
start over. Sorry.
Do that introduction again. Hello, everyone. Welcome to the Botany Collection Tour here at the Royal BC Museum. So I'd like to start off with a territorial acknowledgement first. The lands that we are on are the lands of the Lekwungen people, also known as the Songhees and Esquimalt nations today. So archaeologists would tell us that Indigenous presence in BC dates back to about 14,000 years ago, but oral histories from many nations points to presence here for much, much longer than that. So I'd like to bring that awareness into this tour as we're going to take a look at all of the beautiful plants that have existed in BC for thousands and thousands of years. All right, so I'd like to do a bit more of an introduction. My name is Charlotte. I work at the Learning Center here, and I'll be your tour guide. Behind the camera, we have Rachel and Jenny, and they will be helping me out with tech support. So if you have any questions at any point throughout this tour, don't hesitate, don't keep them towards the end, just throw them in the chat box and they'll be able to get those questions to me, and I will answer them to the best of my ability. All right, so where are we right now? We are in the Fannin Tower on the 3rd floor of the Collection Tower at the Royal BC Museum. The Royal BC Museum is situated right at the south end of Vancouver Island in Victoria, BC. So we are in British Columbia today. All right, so right here, we're at the entrance to the herbarium. The herbarium is where the whole botany collection is stored. And what we're going to find inside, or what we could at one point have found inside, is a collection of 230 to 250,000 plants. So that includes vascular plants, mosses, lichens, tree cones, and things of that sort. So it is a huge collection. But there have been some pretty significant changes to the collection in recent times. So if we look over here, you'll notice a lot of styrofoam, a lot of boxes, and packing materials. If you had walked in here last year this time, you would have seen towers and towers of gray cabinets filled with the botany collections. But now you can see all the way straight through to the window. And that is because the collection and all of the collections in this tower are being packed up to be transferred to the new Park campus, which is a new Royal BC Museum campus that is going to open up in Colwood. And what this campus is a collections, research, and archives center. So it will host all of the collections that now live in the Fannin Tower. So that's what all of this business is going on here. And that is partly why we wanted to deliver this tour to you today, because this is something really unique. We won't be able to see this space as it was used for a very long time very soon. So we wanted to document what it looked like and share this knowledge with you before it changes. All right, so we're going to get into the actual process of how plants and why plants are stored in the botany collection. Over to my right, we're going to talk a little bit about this freezer and its role in the collection. So what happens here in the collections tower is folks will go into a field season to collect new specimens almost every year. And the botany team, they will go out into the field. Right now, they're focusing their efforts into alpine regions because historically these have been the most difficult areas to access. So now that we do have the resources, they'll get into helicopters, go into the mountains, and collect for 2 to 3 days there. So they're hiking, they're camping, and looking for plants. I've been told what they do is they go out and they take one of every plant they see, and they'll put it into a Ziploc bag and bring it back to camp with them. At camp, the next very important part of the process happens, and that is over here. So right up on the top of the shelf is a plant press. So when plants are stored, they need to have all of the moisture removed from them. If any moisture is left in the specimens, they will degrade very quickly. So what happens is they will take the specimens out of the Ziploc bags, they'll place them on a piece of newspaper, fold that over, layer it in with foam and cardboard, and then they will put it in between these wooden blocks and strap it down. Now that process, the cardboard and the newspaper, they are moisture wicking, so they get rid of all the moisture in the specimen, they preserve its shape and its color. So that happens out in the field, and then it all gets brought back to the museum, and it will land in the main floor of the collections tower. But before anything can come upstairs, there is an inspection process. So what are some of the things that scientists or the botany team is likely looking for when they are doing this inspection. Some of the things are chemical and mold concerns, but really what we're looking for is bugs. So bugs are a huge threat to the collection. They love to eat things. They love to eat dead things specifically. And we have a lot of dead specimens in this tower. So it's very, very important that this process happens. So they'll have a visual inspection, and then they will be put into a freezer like this one. We have one up here in the collection space. And they'll be frozen for up to 2 weeks. Sometimes they're frozen, then they're taken out to thaw, then they're frozen again. And what that does is just ensure that any bugs that are hitchhiking on there are killed before it comes up into the collection. It's very important for the safety of all of the rest of the specimens already in here. All right, so that is all for right here. We're going to go to our next stop on the tour. We're going to go down and check out how these plants are processed and put into the collection in an organized manner.
Right. So one moment before we get to this part of the tour, Charlotte, we have a question that I saw pop up in the chat. Jenny, do you mind repeating that question for everyone?
Yeah, for sure. We have a question that is, how do they not mold? So when they're in the plant press, how do they not mold?
Oh, that is a great question. So from what I understand, they will get rid of almost all of the moisture in them, which is what would cause them to break down. So that would be preventing mold from growing. But Heidi, do you have any more specifics?
I don't have.
Oh,
Yes.
didn't work. Go ahead.
Okay.
Yeah.
We actually have a great plant dryer that we take out of the field with us, which is really good at drying them quickly. And that's the key to keep things from molding or losing color or just looking crappy, is to dry them quickly and keeping them dry. So that is the main way to keep mold away. Yeah.
Thank you.
All
Okay,
right.
just a quick introduction. This is Heidi Guest, the collections manager, and we'll be talking a bit about her role in a moment. But first, we're going to be talking about the curator's role. So the curator of the collection is Ken Marr, and he does a lot of the work identifying the plants. So he's done this work for a very, very long time and can identify 85 to 90% of plants just by looking at them. But if not, there is taxonomic keys and reference guides that can be used to get an identification. Once the identification is confirmed, they'll get a number attached to the specimen, and that number will be put into the database online. It is very, very important that that number gets attached to the plant, all the information that was collected with that plant, and put into the database, because otherwise the specimen won't have very much value. If we don't know where it came from, when it came from, we can't use it in any research going forward. So that information will all get grouped together, it'll be put into the database, and that way it can not only be accessed by projects and researchers at the museum, but it can be loaned out to other institutions, and that information along with it can be used by other researchers as well, which is a big part of the work that happens here. So the next part of the process is getting these specimens from those plant presses all grouped together into neatly organized cabinets. All right, so we're going to turn to Heidi Guest, the collections manager. She's going to tell us a bit about this process. So just to start, what is the role of a collections manager in a collection like this?
Well, basically to care for the collection and keep it safe, and also to help make it accessible to the public through the database online, and also for people who come visit, but also for researchers and other experts. So the care involves just making sure things are safe from bugs in particular, but also moisture and light, and keeping things organized. If you lose one specimen being put in the wrong place, because there's 250,000 of them, it can take years to find that specimen. So it's really important to keep things in correct order. Also, with regards to making the collection available, we do have all herbaria around the world have loan programs where we will send stuff out to other institutions for them to study or work on. Often when we get them back, they've done some work on them for us, rename things or change names, identifications, things like that. Also, we have a whole bunch of volunteers. We have about 20 in here right now. Before COVID, we had actually more than that, but we have 20 people that come in every week and work on things like mounting, also imaging station over there. I'm just taking down the mounting that was done yesterday, so you can see some of the beautiful work they do. It's nice. They really enjoy it. It's like a nice artistic, relaxing task.
Yeah.
So
You can come up close and see some of these specimens. That would be great.
I thought this one was really nice. Very artistically presented.
Yeah.
Yeah.
So if you'd like, would you be able to talk about the process of what happens between in the plant press to in the cabinet?
Sure. And you gave a lot of good points on the line, but of course, once we take them out of the press, we freeze them here. Then we basically will go through, check the identifications, re-identify if necessary. Then we put all the data, which was collected when we collected the plants, into the database. And through that, we can create these labels. So each plant has its own label.
Yeah, and we just got a question of what this plant was.
This is a type of sedge, which is a grass-like plant, but not a grass.
Okay.
And it's a fairly common alpine sedge called Carex lacaniae. And it's, pretty cool. So all the data, of course, is going into the database, so it is available online, but also a system to produce these labels from it. So the labels are then put with each specimen, and then our mounters will take the specimen, show you, but this is how it would arrive after we put the label with it and Ken has checked it out. So it'll just be in this paper. Normally we use blank newsprint, but sometimes we run out in the field, so we have to get more paper. So this is what happens. So this is how the specimen would end up before they mount it. So sometimes they have to clean out some of the soil. There's bits of moss in here too, you might see. And of course the labels with it. So they will clean them, and sometimes they won't mount everything that's here because it's just more than we need. So we'll actually save some of them, and we'd call that a duplicate, and we will exchange with other institutions. So I basically accumulated up all the duplicates that happen. So we have a little bag down here that we fill up with the duplicates, and every few years I will go through them all and basically send out a notice to other institutions. We have all this stuff. Would anyone like it? And then package it up and send it out. So I did that a few years ago, and it was many years worth of duplicates, and it was huge. So so that's that. And then they'll decide how they're going to present it. They use this is a methylcellulose glue, and they will glue. There's various methods you can use, but generally you put some glue behind, and then you can also use this linen tape. So it comes in a roll like this. You'll cut a strip, and that you can maybe see here. It's been used here as to hold things down.
These tiny little strips.
Yeah. And I think we had a question. What are the wood blocks used for on the table here?
Are used as they're mounted and glued down. We basically create a stack, and I was just taking the stack down when you come over. So the wood blocks keep things separate so that the glue can dry, nothing sticks to each other, basically. So and here's an example of the weights as well. So when they glued something, they will put a weight down to put it in place.
from, make sure it's actually going to stick where it's supposed to stick. Yeah. See, there's always one. Yeah, we glue the labels on just at the top. You'll see. So we always leave it like that. And that's so that when you're inside a folder full of plants, it's easier to pick just one up. If you're trying to get them apart sometimes with your fingers, you can just go like this and you can. So that's why we do that.
I think there was a couple other questions in the chat if we want to pause and do some questions, if that's okay.
Sure.
Yeah. Hey, Jenny, do you mind reading a couple of those questions out? I saw them pop up and disappear.
Definitely. No worries. So we have about 2 more questions. One was, do the plants get frozen before going up there? Just because you showed that there was some soil. So do they get frozen before they come up to the tower?
Yes. Yeah. They get frozen at least a week. And usually maybe even more. But as soon as we come back, they get frozen. Once we know for sure that they're absolutely dry. Generally, we know they're dry in the field, but you never know. If we've had wet weather, we might just double check. Anyway, they get frozen. Everything gets frozen before it comes into the tower, or enters this room. If it hasn't been frozen, it goes in our freezer. And actually, after the mounting has happened, we freeze them again for a week. And that's just because sometimes these have been, these are from 2021. So they've been sitting around for 3 years because it takes, we're that far behind basically. But yeah, so we just freeze them one more time, then we image them, and then we throw them in the collection. We put them gently into the collection.
And can you expand on why we freeze them?
That is basically to get rid of insects. And that is, as Charlotte was mentioning, that can be the worst problem we could run into. So coarse plants are going to have, I found one yesterday that had just come out of the freezer and there was a dead earwig there. So it had survived being dried and had come all the way from wherever we collected it. And the freezing obviously got it. So that it's a very, very deep cold, which kills all the insects.
And the final question is, what type of paper do you use when you're putting the plants on, like permanently? I see there's a large white paper. Is there a specific type that you use?
Yeah, this is it's got some stiffness to it. It's rag cotton, and it's archival quality. So it's made special for herbarium specimens, and they generally come in different weights. And there's, won't go into too much detail, but that's what that is. Yeah, so it's slightly heavier than your average paper. And we also have these different types of envelopes that we use too. Like you can see here, sometimes a bit of extra stuff has fallen off or is going to fall off, so we put it in the envelope. And this is sort of unfolds like that, so you can see there's a couple of extra bits and pieces in there. And that's also archival quality paper. So everything we use, the glue, the labels, the envelopes, the labels are printed on archival quality paper, so they'll last. Yeah.
Thank you.
Wonderful. That's all the questions.
Great. My last question for Heidi was, how are things organized in the cabinets? You mentioned that if you misplace one specimen, it could take ages and ages to find it again.
Yeah.
So I imagine it is very important to have a specific order for that.
Yeah. So plants are divided taxonomically into various groups. Broadly, we have some non-vascular plants, which would be the mosses, and algae. We don't really have any algae, but we have mosses. Then within the vascular plants, we have different groups, non-flowering and flowering. We have, so in the non-flowering range, we would have ferns, horsetails, conifers, things like that. Within the flowering plants, there's 2 major groups, which I don't think I'll get into. It's a little esoteric. But within each of those groups, they are divided up by family. So there's the hierarchy of taxonomy. It differs first by discipline, but in botany,
The family is important, and then from below that is the genus and the species. So within each of those groups, they're divided up into families, and we arrange them alphabetically by family. And then within the family, they're organized alphabetically by genus, and within the genus, alphabetically by species. Once you get down to species level, they are organized chronologically. So the oldest ones first, and as we gather more, they get down. And we'll have sometimes several folders worth for each species. So folder number 1 will have the oldest specimen, folder number 20 or whatever will have the latest ones.
Yeah, great. Thank you so much for answering those questions. Were there any other questions that popped up?
I didn't see any, but Jenny, is there any more questions? Oh, it doesn't look like it.
All right,
So,
great. We'll continue on then.
Yeah.
Let you go back to your work.
Okay.
So what we're going to take a look at next are a couple of botany specimens that have been selected as great examples of why we have a collection. So we're going to take a look at 4 of those stories today and learn about when they were collected and how they're helping a collection today. So we'll go over farther back in the deepest corner of the collection. So the first one we'll take a look at is right here, and this is Scotch thistle. Now this is a very, very old specimen. You can see here on the label, it was collected in September 26th, and that is 1897, not 1997. So very, very old. And Rachel, if you zoom in on the top there, you can see that it's still got great color left in it. It's very well preserved. So this was the first documentation of Scotch thistle in this area. And Scotch thistle is an invasive species to BC. It is not a native plant. So this documents how herbariums can document the arrival of invasive species. And that's really important if we want to compare what is happening in those ecosystems to what was happening in the past. We can see how aggressive these have been to the natural native plants in the area and make those kinds of connections. So it's very important that we have specimens that go far back in time. The next specimen we'll take a look at is right here, and it is a pale poppy. This one was collected in 2005 by the botany curator Ken Marr and also Richard Hebda. Now, the pale poppy was very rare to find in BC, and it occurs only in rugged alpine habitats in the north. But recent collections of this species, more recent, by Royal BC Museum botanists, document that it is way more widespread and occurs farther south than was previously known. So this exemplifies how it is important to have those older specimens, as well as continuously collecting new specimens, to have something to compare it to and relate it to. We can see that the environment where this pale poppy can grow has changed just between 2005 and now. And this is another example of a specimen that had some extras come with it. So you can see this little pouch here has petals written on it. So inside it will have some petals that have fallen off. Sometimes these will hold seeds and other things like that. Okay. We're going to move on now to the next specimen, and this one is mountain sorrel. Now, this one was collected in 2006 by Ken Marr and Richard Hebda. And what is interesting about this one, it is involved in a new scientific area of research called phylogeography. And that is where botanists can actually look at the DNA sequences of plants, and they can determine the population location and movement during the Ice Age. So this is taking a look back at where these populations could have started, where they migrated to, and they can infer that information from analyzing the DNA of these specimens. So I think that is very, very cool and an interesting new area of the botany field. Okay, the last specimen we're going to look at is right here. This one is a type of grass. It is Romer's fescue. And this one is a type specimen. So I'll talk for a minute about type specimens. A type specimen means that it is the first of that species to be found and identified as a new species. So when that happens, scientists have to write a scientific paper, put it out into the community so that everyone knows there is a new species that has been found. It could be contested if there was competing ideas of what it was. And then those type specimens are most often stored here at the museum. They don't get loaned out like other specimens would. They stay in a cabinet, and we have about 70 type specimens in the herbarium here. So this one was found in a Garry Oak Meadow ecosystem. So that's a very unique ecosystem on Vancouver Island, and you really don't find it very many other places in the world. It is very unique to this area. And what you'll find there are, of course, Garry Oak trees, camas flowers, daffodils in the springtime, and also this kind of grass. And it was the first time that this kind of grass had been collected and documented in a herbarium, and so it became the type specimen for that. And this was collected in July of 1977. All right.
Yeah.
So
I think I saw a
question.
question pop up about these plants.
Yeah.
Jenny, do you mind repeating that one?
Yeah, so the third specimen that we looked at, it looked like there was a bunch of different pieces of the plant. Was that from one plant or just the similar plant from around it? Or do you have any more information about that? Because it looks like a bunch of plants together.
It does. You're right. That is a great question. I'm going to actually go back to Heidi and ask about this. So when you're collecting a specimen in the field, do you collect a few different ones all from the same area, or do you just take from one plant?
Generally, it kind of depends. Like if you're doing a scientific study and you want to make sure that you've got the DNA is all going to be the same plant, then that's important. So this one, because this was part of a phylogeographic study, it would be important that you'd have the same individual. However, certain plants are also, they kind of grow, they can cover a fair amount of ground and still be the same individual. But often collectors will, they'll take, for example, some little tiny plants, you want to take a few to make a decent collection, decent specimen. But yeah, generally you try and get it all from one plant. Yeah.
Okay, great. Thank you. You're welcome. Okay, so now we're going to talk a bit about why we have collections and their importance. So we've covered a little bit about this and what we've already learned today so far. One of the important reasons is because we can loan them out to other institutions. So it acts like kind of like a library. You can check out the specimens that you want, and you can get some information and research from them, and then you can send them back. So that is one of the initiatives that we have here. Other reasons, of course, include climate change. So when we are trying to understand what the climate has done to our province and to our planet, it's important to be able to look back in time and look for any changes that have occurred. So we're very lucky that we have a collection that dates back so far, back into the 1800s, but it's equally important that we keep collecting now. So we can't just say, okay, that's enough. We've seen enough of BC and its plants. We have to continuously keep collecting in order to be able to compare the new information back to the older information and see what changes have taken place. So that is one of the most important reasons that we have the collection here. And that leads me into my next point, which is part of the reason why we have the new park campus. And that is because we were running out of space. Again, if you come in here last year, the cabinets in here took up almost all of the space, and they were almost completely full. And so being in the new park campus will allow the botany team to have a lot more space to work with and proper resources and everything. And one of the other cool things is that we're going to involve the public more in the work we do there. So these kinds of things, bringing the public into the collections and research, will be more available at that campus. I'll just give you a peek into one of the cabinets. We've done a lot of talking about what it looks like in here, but this is what it looks like inside. We've got lots of plants all lined up. If you want to view something, you can pull this out. Take a look, and this is the these will be in order like Heidi mentioned before, chronologically and alphabetically, so we can take a look just at some of these inside here, and many plants all of the same species will be in the same folder here. So as you flip through, dates will change. Generally, they'll get later and later. So we have 1931. Underneath, we have 1953. Underneath that one, we have 1975. All right. Okay, and the last point that I like to mention on this tour is why do we have such a big collection? And I already touched on this a little bit, but I have an example that I like to use, and it has to do with aliens. So if aliens came to Earth and they took a look at humans and they wanted to understand humans, and they just picked up one person and said, okay, we know everything there is to know about humans now. Would that be a very fair judgment? Would they have all the information that they need? No, they likely would not. So it's the same rule that applies here in the herbarium, but also in all the other collections. We want to collect as widely as we can to get a really thorough snapshot and understanding of the province over large periods of time. All right, so that just about wraps up my tour. Thank you so much for joining. Were there any questions before we end things off here?
Yeah, it doesn't look like we have any
Okay.
at the moment, but I think we're right at time. So if anybody has any last minute questions, please type it out in the chat now before we sign off here. Yeah, and then also we do have a couple in-person tours still going this summer. Do you remember the dates for those, Charlotte?
Yes, so Botany, well, you could come see the Botany collection in person if you want to on Thursdays. And I believe we also have a behind the scenes infrastructure tour on Saturdays and Sundays through to the end of August. So if you're interested more about the building and more changes that are happening just beyond the Botany collection, you can come and check that out as well.
Excellent. While we're waiting to see if there's any more last minute questions or comments, I have a question for you, Charlotte. How many specimens do we currently have in the Botany collection right now?
It is about 250,000, but do you mean what is left over beyond prep packing roof?
I meant in general, like how big is it?
Yeah, 250,000.
Okay,
Yeah,
wow,
and
that's
they'll
quite a
bring
few.
Bring in about 1,000 new specimens every field season. I believe that my number is correct
Okay,
there, so.
yeah. I think I saw a question popped up, and that is, are the cabinets fireproof?
That's a great question. And
Yeah.
I think the collections manager again would know
Is she hiding
well.
for a cup?
Well, they're definitely waterproof. And I'm sure they're not absolutely fireproof, but they would probably, you know, hold off the flames for a while.
Right.
Well, that's good. That's good to hear.
And full of paper, that should be good.
Yeah.
Yeah. So I'm not sure of that aspect of them, but yeah.
Yeah. Interesting.
Until they start melting, and then you'd be
Yeah. But waterproof is probably the main priority,
Yeah.
hey, for the mold and all that fun
Yeah.
stuff that would form.
Yes,
Interesting.
Yeah. Definitely.
All
Yeah.
right, cool.
And of course, they keep bugs out too. They're supposed to. They do. Kind of like if you put them in there.
Yeah.
Yeah. Right on.
Great.
All right.
Okay, thank you so much. We'll end our live stream there. Thanks for joining us.